Okay, guys, jumping back in. We're going to finish off this sensations lecture. Hopefully, shouldn't take too much longer since we got about halfway through already. As soon as I get it pulled up, we'll get going. Okay, here we go. So, picking up where we left off, we finished talking about the structures associated with the tongue, that allow us to sense the taste, our taste buds, the sensory cells, and whatnot. So, ready to talk about vision. But before we talk about the eyeball, we'll talk about some accessory structures associated with the eyes. There's a lot that goes into just vision. Aside from your eyeball, we have our eyelids, eyelashes, eyebrows, lacrimal, or what we call the tearing apparatus, and then 6 extrinsic eye muscles we'll talk about on the next page. A lot of these, we're all very familiar with what eyebrows and eyelids are, but they do have a function other than just, needing to be, plucked and whatnot, and, manicured. If that's the correct word, I don't know. But anyway, eyebrows, they provide for facial expression. You can raise your eyebrows when you're excited. You can, bring them down when you're mad, that kind of stuff. But they also prevent like sweat from just like dripping right down into our eyes. So they help with preventing perspiration from getting in our eyes and, other things as well. Your eyelids, the fancy term for those are palpebrae. Singular form of palpebrae would be palpebra. So if we're talking about one, it'd be a palpebra. But the function of those is to block foreign objects, protect from excess light, help with our sleep, obviously, because we close our eyes when we go to sleep, typically anyway. And then we blink. I don't know what the official number is on average, but it's a lot. Every time, we're doing stuff during the day, we're blinking pretty much periodically to help moisten our eyes because our eyelids act kind of like a windshield wiper, distributing that fluid that keeps our eyeballs from drying out. As far as comparing the two, the upper eyelid is a little bit more movable than the lower. In its superior region, it contains the levator palpebrae superioris muscle, which is something I pointed out back when we talked about muscles for spring break, before the world collapsed. But that does exactly what it sounds like. Levate means to elevate or bring upwards. Palpebrae, we know what that means now. Superioris, it's going to elevate our superior eyelid. The space between the upper and lower eyelids is called the palpebral fissure, which we can see here. It's represented with this kind of bracket. Okay, obviously that is going to change based upon how open our eyes are. The angles where our eyelids kind of come together include what we call the lateral commissure, which is over here towards the temporal bone, which is over here on the side of our skull. And then the medial commissure is over here by our nose. And that contains a structure, a fun term to describe what we call our lacrimal caruncle. Now, the lacrimal caruncle is a small reddish elevation, contains sebaceous, so those are oil, and then sudoriferous, which are sweat glands. So it kind of creates a kind of oily, sweaty substance. And the whitish material that sometimes collects in our medial commissure comes from these glands. And that's, what we kind of pick out, kind of crusts up in the morning after we sleep all night. We have associated with our eyelids tarsal or meibomian glands, which secrete a fluid that keeps our eyelids from sticking together. And they're found in the tarsal plate, which is a tarsal plate you can't really see here, but that's what the eyelashes grow from. And if you get an infection, well, I'll just wait on that. And let's talk about the eyelashes. They help protect from foreign objects and perspiration, direct rays of sun. Sebaceous glands at the base of the hair follicles of the eyelashes, what we call sebaceous ciliary glands, releases lubricating fluid into the follicles. And here I'll talk about the infection. If you get infection in the base of those, associated with those sebaceous glands, you can get a stye, which is kind of like a pimple, but on your eyelid, and it's super painful and very unattractive. You can Google what they look like if you've never seen one or had one yourself. They're not very pleasant to experience or have or see. So yeah, let's talk about conjunctiva. Conjunctiva is a term that describes a transparent mucous membrane, covers the inner surface of your eyelids and the anterior surface of your eyeball. So covering the surface of your eye is conjunctiva. And if you flip your eyelids inside out, there's also a conjunctiva there. So there's 2 different types of conjunctiva. Okay. Both of them help to protect the eyeball from drying out. The layer that covers the inner surface of our eyelids is called the palpebral conjunctiva, which makes sense, palpebral or palpebrae, which is our eyelids. and then bulbar conjunctiva, which is what covers the surface of our eyeball. Okay, imagine some of you have probably heard the term conjunctivitis, which is a fancy word to describe pink eye, and that's where this layer of membrane gets infected, and we all know what pink eye is. Over on the right side of the page, we show the structures that collectively make up what we call the lacrimal apparatus, which is a series of structures, some ducts, some tubes, and then a gland which secrete our tears. Okay, lacrimal fluid is just a fancy word for tears. Predominantly is water, but also contains salts, mucus, and even an antibacterial enzyme called lysozyme that can even kill bacteria that grows on the surface of our eyes. Okay. The location of our lacrimal glands is superior and off to the side, so superior lateral to our eyes. And there are a couple of what we call lacrimal ducts, or you'll see them listed as extracorporeal lacrimal ducts here, which drain the tears onto the surface of our eyeball. And this is taking place all the time, even when we're not, in that crying state. Okay? And that keeps our eye just from drying out, as I've mentioned. If we have an emotional response, or if we get something really painful happen to us, we produce more and more of these. And in some cases, tears will flow down our cheeks. Okay? What also takes place is the tears will flow across the eyes and into a superior and into an inferior lacrimal canal that leads to our lacrimal sac. Lacrimal sac is found in the depression of our lacrimal bone, what we call the lacrimal fossa. If you think back to when we talked about the various bones of the body, we learned that there were two little fossas, two depressions, one on each side of our nose on the inside kind of medial portion of our eye socket. That's where that lacrimal sac is contained, and that's where the tears collect. And then from there, they lead down through the nose, or through the nasolacrimal duct into the nose, and that explains why your nose is going to run whenever you're producing lots and lots of tears. Now, on the next couple slides, and 2 slides, I guess, we'll get into talking about the structures associated with the eyeball that are labeled here, which includes the white of our eye called the sclera, the colored portion of our eyes, what we call the iris, the dark center of that, what we call the pupil, but you can see all those other accessory structures I've listed off so far here on this picture and on this picture. Now, our final accessory structures of the eyes are the extrinsic eye muscles, which are what move our eyes in almost any direction, providing the finest motor control in our body. It allows us to very precisely pinpoint where our eyes go, which is very important for doing things like reading the notes that I'm going through right now, or to focus on a ball that's coming at your face as somebody throws it to you, or something like that. Okay. There are 6 total, 4 of which are what we call rectus muscles. You think back to when we talked about muscles of the body, we learned that the word rectus means straight. Okay. And then oblique muscles, there are 2 of those. They are going to allow for rotational movements of your eyes. Okay. Now, we have a lateral view here of the eye socket and the eyeball and those extrinsic eye muscles attached to the eyeball. This is a view where basically the side of the skull has kind of been removed. So, your temporal bone, probably a little bit of the zygomatic bone, maybe even a little bit of the frontal bone has been removed, so we can kind of see the internal features. Okay. On top of the eye and connecting and attaching to the sclera is the superior rectus. And if you contract that, it's going to allow you to look up. And opposite of that is the inferior rectus, which attaches to the bottom side of the eye and as well allows you to look down. Okay. Medial side, on the nose side, in this case of our right eyeball, would be the medial rectus, and this is what allows you to look towards your nose, or if you contract the medial rectus of both eyes, it'll allow you to go cross-eyed. And then the lateral rectus is on the lateral side. It's what allows you to look towards the side of in your periphery. If you contract your right one, it's going to make it look to the right, vice versa. Okay. Now, also associated on the top of our eyeball is the superior oblique. Superior oblique is kind of unique in that it has kind of a V shape to it, or even more of an L shape, I guess. It comes from behind the eye towards the front, and then it hits this kind of pulley-like structure. This hook is called a trochlea, and then allows it to kind of take a right turn. Okay, and then it attaches kind of to the lateral side of our eyes. Okay, whereas the inferior oblique just runs from kind of the nasal side of our eye socket across underneath so it doesn't involve anything back here. Okay. Now back here, surrounding the optic nerve, which is cranial nerve number 2 that carries vision sensations into our brain, is a structure called the common tendinous ring. Okay. Common tendinous ring is where the 4 rectus muscles attach together. Let me see where I can find that somewhere. Okay, here it is. So superior oblique originates posterior. Nope, that's not it. Sorry. Where did I have it written? Well, never mind. I guess I didn't have it written. Oh, there it is. 4 recti rise from a tendinous ring in the orbit, insert into the sclera of the eye. Sorry. Superior oblique does not attach to that tendinous ring, but it's close to it. And like I said, it comes around that pulley-like trochlea and then attaches to the side of our eyeballs. Okay. All 6 muscles attached to the sclera of our eyes, the whites of our eyes, and we'll talk about that on the next page. Okay. I like this set of images at the bottom, kind of give you a feel for the types of movements and what muscles are going to help with those movements. It's not something I would ever ask you to kind of focus on. And this is a sagittal section, so the eyeball's been cut in half. You can see some of those inner features that we'll talk about on the next page. But it also shows the superior rectus, the inferior oblique's been cut. It even points out and shows that levator palpebrae superioris muscle, which is what pulls our eyelids open, the upper eyelid that is, to allow us, to see or to open our eyes when we wake up. Okay. Not something, that I would ask you to know, but there you go. All right, let's actually get into the eyeball. Okay. Even though they're very small, only about 2.5 centimeters or 1 inch in diameter, they are super, super complicated. Only about 1/6 of what we see is I guess we can only see about 1/6 of it. This would be a better way to see it. The rest of it's recessed back and protected by our orbit or eye socket. And 3 layers make up the eyeball, okay, or its wall, that is. The fibrous tunic is the superficial avascular layer, which means superficial, it's on the outside, and avascular, that it does not receive any blood flow. And there's 2 parts that make it up. Okay? And we can see it down here in our illustration, very important, very complicated looking image. This is a superior view looking down at the inside of the eye because the top of the eyeball's been cut away. Okay? I've already talked about the sclera quite a bit. This is what forms the whites of our eyes. Okay? That's one of the 2 structures that make up the fibrous tunic. Okay? It's a layer of dense connective tissue made up mostly of collagen fibers and fibroblasts. And its function is to give shape to the eyeball, make it more rigid, protect its inner parts, and as I mentioned on the previous page, serve as a site of attachment for our extrinsic eye muscles. And the cornea is the last and smaller portion of the fibrous tunic. This is a clear membrane that we see through. Okay? It's what covers the iris and helps focus light into our retina. Okay? And if I'm correct, I believe that's the most frequently transplanted portion of the body when a person donates their organs after death. I believe I read that somewhere once upon a time. Okay, deep to the fibrous tunic is the vascular tunic. Right away, based on its name, should tell you that there's going to be blood flow going to it, because the word vascular is in there. It's also known as the uvea, and it's made up of 3 parts. Okay, let's talk about the choroid first. Okay. Choroid is the layer deep to the sclera. It's also superficial to the retina, so it's in there, it's kind of sandwiched in there between. You see these little red and blue dots. Those red and blue dots represent blood vessels because it's very vascular. Okay? As you can see, highly vascular layer behind the retina provides nutrients to the posterior surface of our retina. Okay? Ciliary body. Ciliary body is going to be labeled here with several parts to it. Okay, very important. Anterior portion of your vascular tunic, lying between the iris and the choroid, extends from the ora serrata. Ora serrata is this jagged kind of structure here shown in kind of a blue. Let's see, jagged anterior margin of the retina to point just posterior to the junctional sclera and the cornea. Parts of the ciliary body include the ciliary processes, which are protrusions or folds on the internal surface of the ciliary body. Also contains a structure known as the ciliary muscle, that changes the shape of the lens, allowing for near or far vision. Okay, so if I zoom in our illustration, okay, here are the little ciliary processes, little finger-like extensions, and they extend off of the ciliary muscle. Okay, now notice connected to the ciliary process are these structures, and they're labeled over here as zonular fibers. Another term for those is a suspensory ligament. They connect the ciliary process to a structure called a lens. Okay? And what they do is they can pull and either kind of flatten out that lens or relax and allow that lens to fatten up a little bit more. And that's what allows us to kind of focus more for far vision or more near vision. Okay? So kind of reading through, this is accomplished by contraction and relaxation of the muscles, which change the tightness of the zonular fibers or suspensory ligaments, which attach the ciliary processes to the lens. Ciliary muscle is a circular band of smooth muscle. Ciliary body also secretes the fluids that fill the eye, and I'll talk about that here after a bit. Okay. The final structure that makes up the vascular tunic is the iris. This is what is colored. As far as, you know, if somebody says they have blue eyes or green eyes, this is what we're talking about. It's suspended between the cornea and the lens, attached at its outer margin to the ciliary process. And you can see it's this portion right here, and the iris would be this right there on each side. There's a space or gap in there between the iris on each side, and that's what the pupil is. And we'll get to that here in just a second. Okay. Now, where does the color of our eyeballs come from? Well, there are melanocytes here, just like there are melanocytes in our skin that we talked about earlier in the semester that secrete melanin. It's what type of melanin, how much we secrete, that determines what our eye colors are. If you have brown eyes, it's because you're secreting large amounts of melanin. Blue eyes, very little. Green eyes have a moderate amount. Okay? And those are the three typical eye colors. Key function of the iris is to regulate the amount of light that enters the eyeball through that pupil, which is the hole in the center of our iris. This is done by contracting the circular muscles to cause the pupils to constrict. This is what happens when there's like a bright light that hits your face, or by contracting the radial muscles, like in darkness, to allow the pupils to dilate. And that's what we're showing off over here on the right side of the page. Okay, the final and most inner portion of our eyeball is the retina. This is where the rods and cones are, if you've ever heard those terms before. The actual photoreceptors, the sensory receptors for vision. There's 120 million rods and 6 million million cone-shaped receptors. Okay, they're called rods and cones because they have a kind of rod or cone shape at the end of the receptor. Okay, that's what's shown down here in yellow. Okay. Now, things are associated with the retina that I want to talk about. The very direct center of our eyeball, right behind the lens, is a region, what we call the macula lutea, which is translated to mean yellow spot. Okay, and this is going to be the direct center of the retina with a structure or a region that's depressed, what we call the fovea centralis, that provides us with the very most perfect and fine vision. Okay? If you think about it, when you read something, you put it right in front of you. Okay, you guys are all looking directly at your page right now, or at the screen right now to read what you are watching or looking at. Okay, when you want to read something, you don't turn your head and look sideways because we can't really make out what's in that vision. Okay, but we do when we are looking directly at something. Okay. Other things, there is a portion of our retina actually where there are no rods and cones. It's this space in here, what we call the optic disc. So any light that is refracted to this area cannot actually be processed because there are no rods and cones here. The optic disc is directly in front of the optic nerve, where the cranial nerve number 2 exits the eyeball, and it does so kind of at an angle, not directly centerized behind the eye like your macula lutea is. Okay. Within the optic nerve, there's a central retinal and artery and vein that bring the blood flow in for the artery and blood flow out via the vein to nourish those different things that we've been talking about. Let's see, talked about everything. All right, let's talk about the lens. The lens is this kind of football shape, I guess, structure. It's behind the pupil, and it's held in place by those zonular fibers, the suspensory ligament. It's pretty cool. I wish you guys would have the opportunity, had we not, had to cancel the rest of the semester in person, to dissect the eyeballs, because it's pretty cool to dissect a sheep or a cow eye and actually be able to pull out the lens. You can actually cut into it and it'll peel like an onion. And that's because there are proteins that make it up. These are called crystallins that are arranged in layers like an onion. Okay. Now, in front of the lens and behind the cornea is a space, what we call the anterior cavity. Okay, and it contains a fluid called aqueous humor. Okay, and that's found in both the anterior and the posterior chamber, which are two different chambers within the entire cavity itself. Okay. Also associated with the anterior cavity is what we call the canal of Schlemm, also known as the scleral venous sinus, which regulates intraocular pressure, so pressure within the eyes. Okay. Now, the bigger and much more pronounced cavity is posterior to the lens and in front of the retina. Okay. And this is a jelly-like substance, and this is probably the coolest thing that you guys are missing out on for dissecting the eyeballs. It's a really kind of jelly. You can almost actually pick it up, and it looks like almost kind of like a really thick snot. That's called vitreous humor or vitreous body, and it's found in this big space, which is called the vitreous chamber. Okay. There's a canal that connects that with the posterior aspect of your lens, and this is called the hyaloid canal, and you can see that's labeled there. Yeah, so that's the structure of the eyeball. A lot going on. Very, very important organ. Think about how important vision is. Okay, let's talk about the ear. Very important sense organ. Pretty complicated as well, which makes sense because it's actually an organ that serves two major roles. Not just hearing. It does have the receptors for sound waves, converting those into nerve signals, allow us to hear and articulate, or not articulate, but to understand the words that I'm articulating. It also contains your receptors for equilibrium. There's three main parts that make up your ear. When we think of your ear, generally we think about what we can see, what we can feel, what we can get pierced. That's your external ear, okay? And more specifically, it's your auricle, or what's also known as the pinna, okay? But the function of that is just to transmit sound waves into your middle ear, okay? It does so by allowing those sound waves to travel into your temporal bone by way of the external auditory canal. Okay, you can also replace the term auditory with acoustic. Okay, this is the ear canal. Okay, basically what you can stick a Q-tip in or stick your finger in, where you have cerumen or earwax that's secreted in there, and we've talked about that all the way leading back to the beginning of the semester. The final structure associated with your external ear is your tympanic membrane. Fancy term to describe your eardrum, which rattles like a drum when sound waves hit it and transmits that vibration inward. Okay. Before I talk about the middle ear, I want to finish talking about and reading through the parts of your external ear. Your auricle, as I mentioned, visible portion of your ear, supported by elastic cartilage. Two parts, principal parts make it up anyway. The helix, which is the rim, okay, and then the lobule, which is your earlobe. What most people, when they get pierced, that's where they're at. Okay. There are ligaments and muscles attached to your auricle, to your head. The external auditory acoustic meatus, also known as canal, 2.5 centimeters or 1 inch tube, leading from the auricle into the temporal bone, ending at that tympanic membrane. Talked about serumous glands secreting cerumen there. Tympanic membrane is a thin, semi-transparent partition between your external auditory canal and the middle ear, covered on the outside by the epidermis, and on the inside it's lined by simple cuboid epithelium. Between them is connective tissue with collagen elastic fibers and fibroblasts. All right, your middle ear is also known as the tympanic cavity. very small air-filled cavity in the petrous portion of your temporal bone. It's only about 2 to 3 millimeters wide, and it's between your eardrum and the oval and round windows, which are structures I'll talk more about on the next page, or actually at the bottom of the page, but more so into the next page, which are openings into your inner ear. Okay. There's also connective tissue between the layers of epithelium. We've seen this picture before, and that was all the way back when we talked about the bones, and that's because there are 3 bones located here in your middle ear. The malleus, which is the hammer, incus, which is the anvil, and then the stapes, which is the stirrup. Okay. When those sound waves rattle your eardrum, this causes a chain reaction where the malleus vibrates the incus, and then the stapes, which is what covers that oval window that I was mentioning just a second ago. And that then transmits sound waves into the structure known as your cochlea. Okay. Let's see. Now, there are a couple muscles I want to talk about associated with these bones. They help limit the motion of the eardrum and limit damage from prolonged loud noises. Fortunately, they're not as effective against sudden sharp noises like a gunshot. There's what we call tensor tympani muscle, attaches to the malleus to help increase tension on the eardrum, and a stapedius muscle, which limits the stapes' motion and is actually the smallest muscle in the body. And I'm not going to talk about, they're so small, I'm not even going to be able to point them out in this illustration. And I do want to talk about several openings that are associated with your middle ear. One of which we've already talked about, that opening from the external auditory canal covered by the tympanic membrane. The oval window, which I mentioned, is covered by the stapes. There's also a round window, another opening into what we call the cochlea, but that's covered by a secondary tympanic membrane. It's kind of like a second eardrum. And then this structure, which is called your auditory tube, also known as your pharyngotympanic or eustachian tube. Okay. This connects your middle ear to your nasopharynx, which is the portion of your throat just inside of your nose, so just posterior to your nose, and explains why if you plug your nose and you blow, your ears pop. Okay. The function of it is to basically equalize pressure in your middle ear, and it will open and close like as we swallow and as we yawn. You notice this a lot, like if you fly and you get the ear popping sensation. It explains why if you chew gum, it helps alleviate that. Okay. That's what this tube, that as you can see says to the nasopharynx, is illustrating. Okay. Now, on the next page, we're going to talk about these structures that make up our inner ear, which include the cochlea, which looks like a little snail shell. That's where the receptors for the sensation of hearing are located. You've also got these 3 what we call semicircular canals. There's 1, there's 2, and there's the 3rd. Okay. And then this area, which for whatever reason isn't labeled, is called the vestibule, which these structures are going to contain the receptors for balance. Now, connected to your vestibule and those semicircular canals is a nerve known as your vestibular branch. Connected to the cochlea is the cochlear branch. Together, those form the entire structure, which is called your vestibulocochlear nerve. And that's one of your cranial nerves, cranial nerve number 8, that connects your inner ear to your brain, travels through what's called the internal auditory canal, which is something I pointed out back when we talked about the temporal bone. Let's talk about that inner ear, also known as your labyrinth, which is because it's kind of a maze-like structure, complicated series of canals. And that's what the term labyrinth means. 2 main divisions of the labyrinth or the internal ear exist. The outer bony labyrinth, which is a series of cavities in the petrous portion of your temporal bone. 3 things make it up. Let's talk about the vestibule that I pointed out here in our illustration on the previous page. Contains receptors for maintaining equilibrium or balance, and is the oval central portion of the bony labyrinth. It would be kind of all in this general area here. Then you've got the cochlea, which was that thing that looked like a snail shell. This is a coiled tube arising from the anterior side of the vestibule. This organ contains receptor cells responding to vibrations created by sound waves, and it contains the receptors responsible for hearing. Open it up like we've done here, you'll see that there are 3 specific chambers within it: a scala tympani, a scala vestibuli, and then a cochlear duct. Okay. The superior chamber is at vestibuli, which ends at the oval window, which I pointed out on the previous page. Inferior chamber is the tympani, which ends at the round window that I pointed out on the previous page. As far as the sense of hearing, the most important is the middle chamber, the cochlear duct, which is full of a fluid called endolymph. And this is home to the organ of Corti, or spiral organ, which is what this kind of reddish shaded structure would be. Okay. This is where you find hair cells, which are the receptor cells for hearing. There's also supporting cells that do the supporting. Okay. At the end of each of those hair cells, there are little hair-like microvilli called stereocilia, which are going to bend when that fluid called endolymph is going to ripple, like what happens if you have a cup of water sitting on your table and you pound on the table and see that ripple effect. That's how sound waves can be converted into nerve signals and allow us to hear, which is pretty cool. All right, the final structures of the bony labyrinth include these three semicircular canals. There's 3 of them containing perilymph. They're named based on the position anterior, posterior, lateral. Not that I'm going to ask you to know that specifically, but they contain receptors just like the vestibule for equilibrium. At the end of each of them is a swollen little kind of bulby-like structure, which is called an ampulla. Okay. Now, that's the outside. On the inside of those structures is an intermembranous labyrinth, which also contains endolymph and contains 2 sacs, which is going to include a utricle and then a saccule, which you can see are labeled here in our illustration. I pointed out on the previous page that each ear is served by the vestibulocochlear nerve, which includes a vestibular branch and a cochlear branch. Okay, that's our video, and that's the lecture on sensations. Now, the great thing is, we, after finally 4 lectures, are going to wrap up talking about the nervous system and move on to a new system, and that system being the endocrine system, which I will start that video and post that just as soon as I can. Thanks.